Okay, today we're going to be revising nucleic acids. Okay, there are two types of nucleic acid. We've got DNA and RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. So, they're both nucleic acids, which is a polymer or a polynucleotide. So if we look at the monomer, and that is the nucleotide. A nucleotide is always made up of three things, 1, 2, 3. A circle, which is a phosphate, a pentagon, which is a pentose sugar, and a rectangle, which is a nitrogen containing base. Okay, so if we start with DNA, DNA has a phosphate, the pentose or 5-carbon sugar, this time is deoxyribose, and it has 4 bases, which are nitrogen or nitrogenous bases. A, adenine, C, cytosine, G, guanine, and T, thymine. RNA is slightly different in its nucleotides. RNA also has a phosphate. The 5-carbon sugar is ribose, and it has 4 bases. It still has adenine A, cytosine C, guanine G, but no thymine. This time it has U, which stands for uracil. Okay, so we need to look at how the nucleotides in DNA join together. So our phosphate attaches to the deoxyribose sugar, which attaches to the base. Now, in order to make one chain, which we call a polynucleotide, the way that it joins up is a sugar-phosphate backbone. So the sugar attaches to the phosphate of the next nucleotide, which remember is a phosphate, a sugar and a base, and then the sugar attaches to the next phosphate. So we can see here that the sugar and the phosphates are joined together, that's the sugar-phosphate backbone, and the bases are sticking out to one side. So that gives us a clue as to how the two chains of DNA attach to each other. Remember, DNA is a double helix. And that's because over here we have bases which are attached to pentose sugars, which are attached to phosphates, more nucleotides. This time we're just going to draw the sugars upside down to make sure the bonds are in the right places. And again, the same thing is true. So it's a sugar-phosphate backbone. So that phosphate will be attached to the sugar of the next nucleotide, which will have a base over here. And then that phosphate will be attached to the next sugar up. And that will have a base. Okay, so the bonds which form between the sugars and the phosphates, they're called phosphodiester bonds. And the bonds that form between the bases, which link the two polynucleotide chains together to make a double helix, they're hydrogen bonds. And there's this very specific set of rules as to how those work. That's called complementary base pairing. And complementary base pairing states that if you have an A, it always pairs to a T, and if you have a C, it always pairs to a G. A and T have 2 hydrogen bonds that pair them up, and C and G have 3. Okay, so we always get that pairing. One of the things that you can look up if you want to do a bit of extra reading is the fact that one of the bases in the pair is called a purine base. It means it's got a double ring. One of them is a pyrimidine base. It means it's got a single ring. And the reason for that is just to keep the helix an equal width apart. Okay, so that's complementary base pairing. What we're now going to do is look at the differences between DNA and RNA, because that's a common exam question. Let's get another piece of paper. Okay, so as we said before, we've got 2 nucleic acids. We've got DNA and RNA. How are they different from each other? Well, the first difference is the sugar. In DNA, we have a pentose sugar, which is deoxyribose. In RNA, it's ribose. The next thing is the bases. Remember, there are 4 bases in DNA, A, C, G and T. But in RNA, we've got A, C, G and U. And the other main difference between them, structural difference this time, is the fact that DNA is a double helix, or has 2 strands, whereas RNA is always going to be single-stranded. And we'll come back to this idea in a minute. But DNA, there's only one type of DNA. There's actually 3 types of RNA. We've got M, or messenger RNA, R, or ribosomal RNA, and T, and transfer RNA. Okay, so they're the main differences there that we've got. So the next thing that we need to look at in this syllabus is how DNA replicates itself. So I'll put that to one side. So we'll look at DNA replication. So as we said before, we've got a DNA double helix. Here are some halogen bonds which between the bases holding the helix together. Okay. So the first thing that actually happens is that the helix has to be separated. So along comes our enzyme. This enzyme is called DNA helicase, and it causes the helix to unwind and unzip. And by unzipping, we mean that we're breaking the hydrogen bonds between the bases. So that causes the 2 chains to separate out from each other like that. Okay, now what we have is we have exposed bases of DNA. So they're no longer paired up anymore. So we could have an A, T, T, C, G, G there. Now just give you a minute to think about what would be along the bottom due to our complementary base pairing rules. So if there's an A there, we'd have a T, A, G, C, C. Okay, so also within the nucleus, in order to allow DNA to replicate, we have 3 DNA nucleotides. There'll be 4 different types because we have 4 bases, but they'll each have, as we said before, a phosphate, the deoxyribose sugar, and the base. So there'll be some adenine ones, some guanine ones, some cytosine ones, and some thymine ones. Now I'm only going to show you the bases, but what will happen is these 3 DNA nucleotides will actually come and pair up over here. So A will pair to T, T to A, to G, to C, to C, and then A, T, T, C, G, G. Now I'm just going to really quickly draw in, although you won't be able to see it, the number of hydrogen bonds. Every time I have a C and G pairing, I get 3. And every time I get an A and T pairing, I just get 2. So complementary base pairing is where the bases join together with hydrogen bonds, but we've not quite got DNA yet because we haven't got the sugar phosphate backbones being sealed together. So we need to draw in those bonds. They're our phosphodiester bonds, and the enzyme responsible for putting the polynucleotide chain together is DNA polymerase, because we're making the polymer again. Okay, now we've got 2 pieces of DNA. All they need to do is twist up, and we'll have 2 identical pieces, identical to the first piece of DNA that we started with. So the type of replication that we've got we call semi-conservative. It's semi-conservative because in each double helix of DNA we have one of the strands, which is what we call a parent strand of DNA, or an old strand, it was there already, but the other one is a new strand. So we have 1 parent and 1 new strand, and that's what semi-conservative replication is, because we've conserved one but we've made a new one. And the last thing that we need to look at in this topic is looking at the DNA's job, which is protein synthesis. DNA codes for a protein. So we come back to this at A2, so we only need to know it in a little bit of detail. So protein synthesis, that's the job of DNA. It replicates itself and it codes for proteins. So in protein synthesis there's 2 stages. Firstly, transcription, which takes place in the nucleus, and secondly, translation, which takes place out in the cytoplasm. So I'm going to show that by trying to show this as the nuclear envelope here. A big nuclear pore there. So inside the nucleus we have our DNA. Again, we're just going to draw it all twisted up like that. The first thing that has to happen is, again, we need our helicase enzyme to come in and unzip it, break those hydrogen bonds. Now we've got some bases exposed. So we use the same bases there. This time though, because we're making messenger RNA, which is single-stranded, we only need to use one of our 2 DNA polynucleotide strands. So inside the nucleus we have 3 RNA nucleotides. So we can make RNA. Some of them have A, some of them have C, some of them have G, and some of them have U. So this time, whenever there should have been a T, we're going to replace it with a U. So here, A is going to pair to a U, T to an A, T to an A, C to a G, G to a C, G to a C. And remember, they pair up by hydrogen bonds. So again, I'm putting in 2 hydrogen bonds between an A and a T or an A and a U pairing, and 3 between a C and a G. We need our chain, our mRNA, to join to itself, our sugar phosphate backbone, and the enzyme that's going to do that this time is RNA polymerase. Now we've got some mRNA, single-stranded, so it's nice and small, so it can exit the nucleus. Out it goes. That's the end of transcription. And it goes to a structure in the cytoplasm called the ribosome. And there's our ribosome. And the mRNA strand will actually attach itself into the groove between the 2 subunits of the ribosome. So here's our strand, U, A, G, C, C. So the way our genetic code works is it works in triplets. It's a triplet code. So that's the first triplet, GCC. Here's the second one. They're called codons, UAA. Okay, so there's the first 3. There's the second. What the ribosome does is it reads each codon in turn. So the first one says GCC. And outside in the cytoplasm we have tRNA. Which tRNA looks a bit like a clover leaf. It's got 3 bases on the end of it, which we call an anticodon. So the anticodon we need here is CGG. And each tRNA is attached to an amino acid. So what happens is the tRNA will line up with its anticodon next to the mRNA codon. So CGG, there it is. And then the amino acid that it's attached to will be deposited at the ribosome. That says pro for proline. The ribosome will then read the second of its codons. This time it says UAA. So that will match with an anticodon that says AUU. So we'll have another tRNA there, which will be attached to another amino acid. I'll just put the number 2 in there. So now we've got 2 amino acids next to each other. All that has to happen is they need to join together, and that bond is called a peptide bond. And now we've got the beginnings of the primary structure of the protein, which is just a chain of amino acids. What would now happen is the mRNA would continue to move through that groove, and we'd look at the 3rd codon, another tRNA would bring another amino acid in, and we would build up our protein, our amino acid chain. Okay, that's your tutorial on nucleic acids. There's an exam question and the mark scheme on the internet for you to try.